friends, welcome back to my channel, and today we are going to study about lipid part 5. In this we'll learn about sphingolipids and sterols. So sphingolipids are the derivatives of sphingosine, like glycerolipids are the derivatives of contain glycerol backbone. Similarly, sphingolipid contain the backbone of sphingosine. So like glycerol, it also has 3 carbons, but the third carbon is fixed. So for the attachment, only 2 carbons are available, that is carbon number 1 and the carbon number 2. With the carbon number 2, fatty acids are attached. So now here in the carbon number 2, like glycerol, it is not CHOH, it is CHNH. So now if you see CNH and C double bond O, NHCO bond, what you will call this bond? Peptide bond or the peptide linkage. With carbon number 1, depending upon the X group, like glycerophospholipids, sphingolipids are also named differently. Clear? So sphingosine, here carbon number 1 and carbon number 2 are available only for the attachment. With the carbon number 2, fatty acid is attached through peptide bond, and with carbon number 1, different X groups are found. Now what are these X groups? So here is the name. If the X group is H, the compound is named as ceramide. If the X group is phosphocholine, the compound is sphingomyelin. Where it is found? Myelin. That means in the myelin sheath. So it is found in the plasma membranes of animal cell, and that too neural cells. Next is glucosyl cerebroside. That means it contains glucose. So this is a glucose unit. That means the bond will be glycosidic bond. So where this cerebroside we can find in the neural tissue, and those with glucose in the plasma membrane of cells in non-neural tissues. Similarly, lactosyl cerebroside, which contains di, tri or tetrasaccharides. Here the saccharides are glucose and galactose. So it is known as globoside. So globoside is also a glycolipid only, because glyco means carbohydrate. So here glucose is present. That's why it is glycolipid. Here also glucose are found. That's why it is also a glycolipid. So here when one glucose is present, the name is cerebroside, and when more than one carbohydrate is present, that is 2, di, tri or tetra, we call it as globoside. Next is ganglioside, when complex oligosaccharide is present and one of the oligosaccharide is sialic acid, that is N-acetyl neuraminic acid. The structure is like this. This is the structure of sialic acid. The name GM, this M stands for mono N-acetyl neuraminic acid. That means only one molecule of this sialic acid is present. If in the structure 2 molecules are present, then we call it as GD. If 3 molecules are present, we call it as GT. So ganglioside GM, ganglioside GD, ganglioside GT. So this GM, GD and GT tells us how many molecules of sialic acids are present in the sphingolipid. Now, sphingolipids at the cell surface, they are the sites of biological recognition. For example, as you can see in the blood group. So what is there in our blood group? Sphingosine is there, fatty acid is attached, and these carbohydrates are found. That is glucose, galactose, N-acetyl, galactosamine, again galactose, and this is fucose. So this is known as O antigen. Now with this galactose, if one more molecule of N-acetyl galactosamine is present, it forms A antigen. If simple galactose is present, it forms B antigen. So these are the sphingolipids which are found on the cell surface and function as blood group antigens. So a person is of blood group A or blood group B. That depends upon the carbohydrates present in the sphingolipid. Now, phospholipids and sphingolipids, these are degraded in the lysosomes. So they are continuously degraded and replaced. So different enzymes are present that can degrade phospholipids and sphingolipids. So like phospholipase A1, which degrade the bond shown here. Phospholipase A2, which degrades the bond of second carbon and the fatty acid. Phospholipase 3, which breaks this bond, and phospholipase D, here it is shown, the bond it breaks is here. The point of its breakage is shown here. So these are the phospholipases that degrade the lipid molecules. If not properly degraded, they get piled up in the cell. That can cause various diseases. Now, in case of gangliosides, it contains complex oligosaccharides. So one by one, all the sugar molecules are removed, finally yielding a ceramide. So there are multiple human diseases that occur due to abnormal accumulation of these membrane lipids if not degraded. So ceramide, that is the basic element, this is cerebroside. This molecule is removed with the help of enzyme beta-galactosidase. If not removed, then it will cause a disease known as generalized gangliosidosis. So after the removal of this molecule, this is remain. Now to degrade and remove this carbohydrate, the enzyme used is hexaaminosidase. If not removed, in that case, it will cause the disease known as Tay-Sachs disease. Now, after removal, what is left? This much. Now, this fifth numbered carbohydrate is removed with the help of enzyme ganglioside neuraminidase. Now, the compound formed is ceramide. Only these two are remain. Now, this is removed with the help of enzyme again beta-galactosidase. If not removed, now the left compound is ceramide with this only carbohydrate. Here, this carbohydrate is removed with the help of enzyme glucocerebrosidase. If not removed, it causes the disease known as Gaucher disease. Now only left ceramide and free fatty acid. Now from globoside, here also the last carbohydrate is removed with the help of enzyme hexaaminosidase A and H. If not removed, it causes Sandhoff's disease. So after removal, the compound formed will be with 3 carbohydrate. Now again, this last one is removed with the help of enzyme alpha galactosidase A. If not removed, it will form the disease Fabry's disease. So after removal of this, it will come to this point. Similarly, from sphingomyelin also it can come. It will also give to free ceramide. Free ceramide will come from here after removal of this last carbohydrate. So this is degraded with the help of sphingomyelinase. If not degraded, it causes disease known as Niemann-Pick disease. The diseases that results due to abnormal accumulation of membrane lipids. Now sterols. They have 4 fused carbon rings. They are the structural lipids present in the membranes of most eukaryotic cell. So what it contains? It contains steroid nucleus. These are 4 fused isoprene rings, a polar head with the 17th position connected with a long alkyl side chain, and these are hydrophobic in nature. Now, cholesterol, among sterols, cholesterol, this is the major sterol in all animal tissues. Similar sterols are found in other eukaryotes like stigmasterol in plants, ergosterol in fungus. Steroid hormones, these are potent biological signals that regulate gene expression. Bile acids, these are polar derivatives of cholesterol. For what our body use bile acids to emulsify fats, help in digestion. So sterols, these are non-polar molecule and form the structural lipid. Now one question from my end for you all, that is most hormones such as peptide hormones exert their effect by binding to cell surface membrane. However, steroid hormones do so by binding to cytosolic receptor. How is this possible? So to answer this, you have to keep in mind that this steroid hormones are crossing the plasma membrane barrier. So what is the property of steroid and what is the property of plasma membrane? If you think you'll get the answer, do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.